Hello and welcome. All right, let's jump right in because today we're talking about one of the most incredible, most foundational processes in all of biology. It's called gastrulation, and trust me, this is where life really starts to get interesting. It all boils down to this one huge question. You start with what is basically a featureless little ball of cells. How in the world does that transform into something with a brain, a heart, limbs, a whole complex animal? That's the amazing puzzle we're going to unpack.
So let's set the scene. Our story begins with the earliest version of an embryo, a structure we call the blastula.
And this is it. This is our starting point. The blastula is pretty much just a hollow sphere made of cells that all look the same. There are no organs, no head, no tail, absolutely no hint of what it's going to become. And that presents the fundamental challenge of development, right? How do you get a detailed body plan from something so incredibly simple?
Well, to figure this out, scientists needed a way to see the future. They needed a map. And what they came up with was a truly brilliant tool called a fate map.
I mean, this is the real head-scratcher, isn't it? If you're looking at this ball of cells, and they all appear identical, how could you ever tell which ones are going to form, say, the skin versus the ones that are going to form the brain?
The answer came from this groundbreaking work by Walter Vogt. His technique was actually beautifully simple. He used harmless dyes to put a little mark on a small patch of cells. Then, he just waited and watched. As the embryo developed, he could literally follow that colored patch and see where it ended up, creating a map of what each region was destined to become.
And this gave scientists a whole new language. When you see a fate map and a region is labeled prospective something, like prospective neural plate, it means we've watched these cells and we know for a fact this is where they're headed. So, a fate map isn't a guess, it's a roadmap based on direct observation.
Okay, so we have a map. But a map is pretty useless if you don't actually go anywhere, right? The process that turns this 2D map into a 3D living organism is gastrulation. This is the main event.
Gastrulation is that journey. It's this massive, perfectly choreographed migration of cells. That simple hollow ball starts to fold, to tuck, to stretch. It's like a piece of living cellular origami. A single layered structure begins to reorganize itself into the distinct inner, middle, and outer layers that will eventually form the entire body.
So we've got the map, and we know the journey has started. But what are the actual mechanics here? How do the cells physically make these moves? It turns out, different animals have different strategies. Let's check out a couple of examples.
First up is a small marine animal called Amphioxus. And it's a fantastic place to start because it shows us gastrulation in its simplest, most straightforward form.
The secret to its simplicity is all about yolk, or more specifically, the lack of it. Amphioxus embryos have just a tiny bit of yolk, and it's spread out evenly. That means there are no big, heavy obstacles in the way of the cells that need to move.
So, because there's no heavy yolk getting in the way, Amphioxus can use a very direct method called invagination. The best way to picture it is to imagine taking a soft, hollow rubber ball and just punching one side in with your thumb. That's it. An entire sheet of cells just flattens and folds into the interior. And just look at what that one simple move accomplishes. Instantly, you have a two-layered structure. You have an outer layer and an inner layer. You've formed a primitive gut and an opening to the outside world. The entire basic body plan is set up in one incredibly efficient motion. But what happens when things aren't so easy? For our next case study, let's look at amphibians. They have the same goal, but their cells have to navigate a major obstacle course to get there.
And this is the critical difference. That simple, elegant punching in that we just saw in Amphioxus, in an amphibian, it's impossible. It just doesn't happen. So, why not?
The obstacle, once again, is yolk. But this time, there's a ton of it. The cells at the bottom are absolutely packed with heavy, energy-rich yolk. It's like they're all carrying these massive backpacks. They're just too big and bulky to do that neat little fold we saw before.
So evolution found a really clever workaround. Instead of a whole sheet of cells folding in at once, individual cells on the surface actively crawl towards a specific spot, this groove called the dorsal lip, and then they just roll over the edge into the interior. It's less like a fold and more like a continuous waterfall of cells flowing from the outside in.
And as this cellular waterfall continues, that little groove expands to form a complete circle. For a little while, you can actually see the last of those big yolky cells filling that opening. forming what's called a yolk plug, right before they too get pulled inside.
So you see, whether it's by a simple fold or a complex roll, the end result of gastrulation is exactly the same. It accomplishes one of the most important jobs in development. It establishes the 3 essential layers of cells that will build the entire body.
And here they are, the 3 primary germ layers. And just think about this for a second. The outer layer, the ectoderm, becomes your skin, your brain, your entire nervous system. The middle one, the mesoderm, forms your muscles, your heart, your bones. And that inner layer, the endoderm, becomes the lining of your gut, your lungs, your liver. Every single part of you started in one of these 3 teens.
This is why this moment is so profound. Every single organ, every tissue in an animal's body has its origin in one of these 3 layers. Gastrulation isn't just about moving cells around. It's about creating the fundamental architectural plan for a living being.
In the end, it all comes down to this number, 3. From a simple, hollow ball of cells, this incredible migration creates 3 distinct layers, each one with its own epic destiny. It's the blueprint for life. So, gastrulation draws up the blueprint. It sorts all the materials into the right piles: ectoderm, mesoderm, endoderm. But a blueprint is just a plan. The next great question is, what provides the actual instructions that tell these layers how to build a heart, or a nerve, or a lung? Well, that's a story for another day.